Hello students, here I am Ashok Waghe once again in front of you to explain you the remaining part of molecular basis of inheritance. During the previous lecture we completed about the central dogma of protein synthesis, and in addition to that the first part of protein synthesis, that transcription we completed. And in addition to that we completed about the introductory part of genetic code, and I explained how to read the dictionary of genetic code. That also I completed. Now today we want to see the second part of genetic code, that is characteristic feature of genetic code, and in addition to that some introductory part of that is protein synthesis part 2, translation mechanism. So now let's start about the understanding about the characteristic feature of genetic code. So before we start about the genetic code, three important definitions I keep in front of you. So they are there on the screen. So number one, that definition that is called as the codogen. Second one, it is called as codon. And third one, it is called as the anticodon. Smallest possible sequence, that is triplet of nucleotides present on the DNA strand, which can specify one particular amino acid. So that's why it is included here thymine also, because it is related to that of DNA. Thing is that when it is transcribed to that of mRNA, so it becomes what you call as codon. The codon is the smallest possible sequence, that is triplet of nucleotides present on mRNA strand, which can specify one particular amino acid. So of course here thymine is replaced by uracil. That is very important point you should note here. Third definition it is there, that is called as the anticodon. It is a triplet of nucleotides present on the anticodon loop of tRNA, which is complementary to the codon of mRNA. So that's why now we want to see here the next one. Actually today we want to see, that is called as the characteristic feature of genetic code, or it is also called as the mRNA language, or it is also called as the cryptogram. Genetic code of DNA has certain fundamental characteristic feature. So number one, genetic code is triplet code. Sequence of 3 consecutive bases constitute codon, which specify one particular amino acid. Base sequence in a codon is always from 5' end to 3' end direction. In every living organism, genetic code is a triplet code. Second one, genetic code has a distinct polarity. Genetic code shows definite polarity, that it is having the direction. It therefore is always read in 5' end into 3' end direction, and not in 3' end to 5' end direction. So very important, that is you should read always from 5' end to 3' end direction. That's why it has a definite polarity, you have to remember. Otherwise message will change from 5' end to 3' end, that is AUG. So that's why you have to remember, you always read the genetic code, it is having the distinct polarity. Number 3 characteristic feature, that genetic code is non-overlapping. So it's a very important one. Code is non-overlapping, that is each single base is non-overlapping. That is each single base is a part of only one codon. Adjacent codons do not overlap. If non-overlapping, then with the 6 consecutive bases, only 2 amino acid molecules will be in the chain. Had it been overlapping type with 6 bases, there would be 4 amino acid molecules in a chain. Experimental evidence is in favor of non-overlapping nature. So that's why this very important one you can remember. So one what you call as amino acid, it is not common in between 2. That's why that is another meaning of the non-overlapping. Next one, it is that genetic code is commaless. There is no gap, there is no punctuation mark between the successive or consecutive codons. Genetic code has degeneracy. Usually single amino acid is encoded by single codon. However, some amino acids are encoded by more than one codons. For example, cysteine has 2 codons, while isoleucine has 3 codons. This is called degeneracy of the code. Degeneracy of the code is explained by Wobble hypothesis. Here the first 2 bases in different codons are identical, but the third one variable. Fourth one, genetic code is universal. By and large in all living organisms, the specific codon specifies same amino acid. For example, codon AUG always specifies amino acid methionine in all organisms from bacteria up to the humans. That's why it is called as the genetic code is universal. Genetic code is non-ambiguous. Specific amino acid is encoded by a particular codon. Alternatively, 2 different amino acids will never be encoded by the same codon. It's a very important one you have to remember well. Next one, it is there, initiation codon and termination codon. AUG always an initiation codon in any and every mRNA. AUG codes for amino acid methionine. Out of 64 codons, 3 codons, that is UAA, UAG and UGA, are termination codons, which terminate or stop the process of elongation of polypeptide chain, as they do not code for any amino acid. Next one, codon and anticodon. Codon is a part of DNA. For example, AUG is a codon. It is always represented as 5' end to 3' end, that is AUG. Anticodon is a part of tRNA. It is always represented as UAC, that is 3' end to 5' end, you have to remember. So whatever these genetic code characteristic features are there, they are very important. We want to see here. Now, as I mentioned earlier, I have to explain you about the few things regarding the what you call as mutation and genetic code. That is always in another language it is called as the frame shift mutation. So while dealing with that of the previous lecture, I mentioned about the frame shift mutation. So first of all, I want to tell you about the frame shift mutation, and then we'll proceed further to this slide, that which you are observing in front of you, that is tRNA, the adapter molecule. So that's why mutation and the frame shift mutation I want to explain you. Mutation is a phenomenon in which sudden change in the DNA sequence takes place. It results in the change of genotype, that is character. Along with the recombination, mutation is a raw material for evolution, as it also results in variations. During mutation, possibility of loss, that is deletion, or gain, that is insertion or duplication of a segment of DNA, results in alteration in the chromosome. Mutation can also occur due to change in a single base pair of DNA. This is known as point mutation. For example, sickle cell anemia. I hope you already completed sickle cell anemia during previous lectures. Deletion or insertion of base pairs of DNA causes frame shift mutations or deletion mutation. Insertion or deletion of one or two bases changes the reading frame from the point of insertion or deletion. Insertion or deletion of 3 or multiples of 3 bases results in the insertion or deletion of amino acids, and reading frame remains unaltered from that point onwards. So in this way we completed here the characteristic feature of genetic code. Now we want to see here the next part, that is called as the tRNA as an adapter molecule. See, whatever the types of RNA are there, you already completed in 11th standard. That 3 types of RNA are there. Ribosomal RNA, tRNA, as well as mRNA we completed, their characteristic feature we completed here. So now in front of you the three diagrams are there. One diagram, the second diagram, that is adapter, and the next one, that is mRNA you are observing. The tRNA, that is adapter molecule, one small part it is there in your textbook. So that's why I want to explain that part. The diagram, it is there in front of you. Please refer the diagram while my explanation is going on. Scientists considered that there has to be a mechanism in which tRNA will read the codon and also simultaneously binds with the amino acid, as amino acid does not have any special capacity to read the codon. So tRNA is considered as an adapter molecule. This role of tRNA was understood much later. Clover leaf structure, that is two-dimensional, of tRNA possesses an anticodon loop that has bases complementary to the codon. See the anticodon that is in the diagram, it can be seen properly. CUC, that I mentioned anticodon in that diagram, or it is already there in the textbook. You can observe that diagram. It is called anticodon. It shows amino acid acceptor end, 3' end, having unpaired cytosine and adenine bases, that is amino acid binding site, to which amino acid binds. Every amino acid there is a specific tRNA. Initiator tRNA is specific to methionine. There are no tRNAs for stop codons. In the actual structure, the tRNA molecule looks like inverted L, that is in the three-dimensional structure. See the second diagram, it is there also in front of you. So that's why imagine L-shaped structure, and that's why this one, it is considered as a L-shaped structure. Now interesting thing, the title it is given that tRNA, the adapter molecule. Why it is called as the adapter molecule, that also I want to explain here. See, to the 3' end I mentioned, the arm it is there, it is related to that of the attachment of any amino acid, that according to the anticodon which is present here, which able to read the codon language. So that's why I shown the glutamic acid at the 3' end. Whereas this anticodon always pairs with the nitrogen base, anticodon always shows the nitrogen base pairing with that of the codon. So in that situation, so it also pairs with that of the mRNA. To one side it get attached to that of the amino acid, and to another side it get attached to that of the mRNA. That's why it shows similarity with that of the adapter which is used in electricity. So that's why that tRNA, it is considered as an adapter. So with this, now we completed the tRNA as an adapter, and in addition to that we completed about the characteristic feature of what you called as genetic code we completed. In addition to that, we want to proceed further, as I mentioned in the earlier, in the introductory part of this lecture I already mentioned, isn't it? So that's why we want to see here second part of protein synthesis, that is called as the translation. In the central dogma I already mentioned, whatever the DNA it is there, it is transcribed to that of the mRNA. That transcription mechanism we already completed. That mRNA, it synthesizes the polypeptide chain, and that's why the protein synthesis it is there. So that mechanism it is called as the transcription. That's why that mechanism it is called as the translation. Hence we want to see here the next part, that is second part, translation mechanism, that is actual protein synthesis. Translation is the mechanism in which codons of mRNA are translated and specific amino acids in a sequence form a polypeptide on the ribosome. All types of proteins are synthesized by the cell within itself. Process of translation requires amino acids, mRNA, tRNA, ribosomes, ATP, magnesium ions, enzymes, elongation, translocation, and release factors. Amino acids form raw material for protein synthesis. About 20 different types of amino acids are known to form proteins. These are available in the cytoplasm. DNA controls synthesis of proteins having amino acids in specific sequence. This control is possible through transcription of mRNA. Genetic code is specific for particular amino acid. You are knowing already about that. RNAs serve as intermediate molecules between DNA and protein. Ribosomes serve as site for protein synthesis. Each ribosome consists of large and small subunits. These subunits occur separately in the cytoplasm. Only during protein synthesis these two subunits get associated together due to magnesium ions. A ribosome has one binding site for mRNA and that is 3 binding sites for tRNA. They are P site, that is peptidyl site, tRNA site, that is A site, that is aminoacyl or tRNA site, and E site, that is exit site. Only first tRNA amino acid complex directly enters the P site of ribosome. In eukaryotes, a groove is present between the two subunits of ribosomes. It protects the polypeptide chain from the action of cellular enzymes and also protects mRNA from the action of nucleases. So that's why I explained how the background of translation mechanism it is there. Now I want to explain mechanism of translation, that is synthesis of polypeptide chain. It involves 3 steps, that initiation step, second elongation step, and third termination step. So that's why I am showing with the help of 3 diagrams that you are observing. Number 1, it is called as the initiation complex. Second one, you are observing elongation 1, and the third one you are observing the termination. And also you are observing how the shifting of the position of the ribosome it is there, that is from 5' end to 3' end in these 3 diagrams. Okay, so that's why we want to see here that how the initiation of polypeptide chain it is there. Activation of amino acid is essential before translation initiates, for which ATP is essential. Small subunit of ribosome binds to the mRNA at 5' end. Initiator codon, that is AUG, is present on mRNA, which initiates the process of protein synthesis, that is translation. Initiator tRNA binds with the initiation codon AUG by its anticodon UAC through hydrogen bonds. It carries activated amino acid methionine in eukaryotes or formyl methionine in prokaryotes. Now the large subunit of ribosome joins with the smaller subunit that requires magnesium ions. Initiator charged tRNA with the activated amino acid methionine occupies the P site of ribosome and A site is vacant. See, remember well here, the first tRNA, it is bringing the amino acid directly go to the peptidyl site instead of going to that of the A site. Remaining other tRNA, they come to the A site first, then they are shifted to that of the P site. We'll see that in the further stages. Second step it is there, that is called as the elongations of polypeptide chain. During this process, activated amino acids are added one by one to first amino acid, that is methionine. Amino acid is activated by utilizing energy from ATP molecule. This amino acid binds with the amino acid binding site of tRNA. This results in formation of tRNA amino acid complex. Addition of amino acid occurs in 3-step cycle. A. Codon recognition amino acetyl tRNA molecule enters the ribosome at A site. Anticodon binds with the codon by hydrogen bonds. Amino acid on the first initiator tRNA at P site and amino acid on tRNA at A site joined by peptide bond. Here enzyme ribozyme acts as a catalyst. At this time, first tRNA at P site is kicked off. That is, that tRNA, empty tRNA leave from this place. C, that is translation, the tRNA at the A site carrying a dipeptide at the A site moves to the P site. This process is called translation. The translation, both the subunits of ribosome move along in relation to that of tRNA and mRNA. Hence, the tRNA carrying dipeptide now gets positioned at P site of ribosome, making A site vacant. At this site, then next charged tRNA molecule carrying amino acid will be received. During this process, first uncharged tRNA is discharged from the E site. This process is repeated as amino acids are added to polypeptide. It takes less than 0.1 second for formation of peptide bond. Third charged tRNA with its amino acid arrives at A site of ribosome. Anticodon and codon bind by hydrogen bond. Polypeptide bond is formed. Second tRNA is discharged from P site to E site and leaves the ribosome. So the events like arrival of tRNA amino acid complex, formation of peptide bond, ribosomal translocation and removal of previous tRNA are repeated. As ribosome move over the mRNA, all the codons on mRNA are exposed one by one for translation. The diagram, it is already there in the textbook. Students, you can refer that diagram also. And the last one process step, it is there, that is called the termination and release of polypeptide. At the end of mRNA, there is a stop codon. I already mentioned about that, UAA, UAG or UGA. It is exposed at the A site. It is not read and joined by anticodon of any tRNA. The release factor binds to the stop codon, thereby terminating and the translation process. The peptide is now released in the cytoplasm. Two subunits of ribosome dissociate and last tRNA is set free in the cytoplasm. mRNA also has some additional sequences that are not translated and are referred as untranslated regions, UTR. The UTRs are present at both 5' end and 3' end. They required for efficient translation process. Finally, mRNA is also released in the cytoplasm. It gets denatured by nucleases immediately. Hence, mRNA is short-lived. Now, the same process which I just explained theoretically, I want to show a video related to that of translation. So let's play the video and let's see how the translation mechanism it is there. See this diagram, it is there from the textbook. I already said please refer this diagram also. See the translation mechanism which I already explained theoretically. Now you are observing in the form of video. So once again I mentioned translation is the synthesis of protein. It requires mRNA, it requires ribosome, larger subunit, smaller subunit, tRNA, releasing factor. So stages of translation: initiation, elongation, termination. Now see the actual translation mechanism. This is mRNA. UTR you are observing. Even this side also UTR you are observing. So the smaller subunit of ribosome get attached. That's why initiation site. Here the tRNA bringing particular amino acid which is represented with the help of red color. UAC, that is anticodon. codon, anticodon, they pairs with each other. That's why the codon it is red. First one, that is methionine, it is brought here. The larger subunit you are observing. Directly come to the peptidyl site. So second one, it arrived at the A site. See the previous amino acid, methionine, and a new amino acid, they are forming the bond here. See the bond formation. That's why the empty tRNA leaving this one. Third one you are observing, again the polypeptide chain formation, it is there. So thus the long chain of polypeptide you are observing here, long chain of amino acid you are observing here, and that's why it finally reach up to the termination end, where everything they are separated from each other. That's why the stop codon it is there, and the mechanism of translation it is terminated, that is completed, or that is stopped here. Thus the translation mechanism it is completed. Okay students, thus we completed about the what you call as the entire protein mechanism, protein synthesis mechanism we completed. The transcription mechanism in which we completed the mRNA synthesis it is there, and in this video just we completed about the translation, that how the amino acids they are linked with each other. So we completed according to the central dogma of protein synthesis. I want to remind you about the central dogma of protein synthesis, that is from DNA, mRNA it is synthesized. That's mechanism it is called as the transcription. And from the mRNA, whatever the polypeptide chain synthesis it is there, that we completed here. Now we want to see how this mechanism it is regularized, that how it is controlled at the cellular level. Because you are knowing every time the proteins or every time the enzymes they are not required. So that's why as the proteins or a particular enzyme it is not required in that condition, this protein synthesis mechanism should be stopped. So this can be controlled at the translational level, or it can be controlled at the transcriptional level. So that's why we want to see here the regulation of gene expression next time. Regulation of gene expression next time we want to see. So students, today we completed about the characteristic features of genetic code, and in addition to that the very important part we completed, that is second part of protein synthesis, that is called as the translation. So I played video also for you people, and in addition to that I explained theoretical part also. But the question arises, every time these cells require proteins or enzymes? No, every time they not required. Then what happened when they not required any enzyme or any protein? That is how this protein synthesis mechanism, or how the role of genetic code or whatever the mechanism of gene it is controlled or it is regulated, the question arises. So this part definitely we want to complete next time. Now we want to stop here only. Thank you very much.